Yeah, so obviously the topic is about the broadest possible for those of us who are involved in clinical research and who aim to see vaccines and drugs emerging against COVID, but also against other diseases that may be competing for attention with COVID and that may present very similar. So I spent some time thinking about this in terms of their breadth of knowledge and their expertise, from microfluidics to diagnostics to really drugs and vaccine development per se, to the really important basic science foundation that make drug and vaccine development possible. So I'd like to talk about this from a bird's eye view, with a strong focus on innovation and on questions that lie clearly between the disciplines and that will haunt us throughout drug and vaccine development, not just for COVID, but also for other respiratory viral infections. I'm a board member with ISERV, the International Society of Influenza and Other Respiratory Viruses, which has been very involved in bringing the scientific community around, not just singular viruses, but the whole picture, because there are a lot of translational questions occurring between them that also directly impact both drug and vaccine development. So without much further ado, I'd like to see how we can, first of all, acknowledge the country where this meeting should be taking place, at least virtually. I found this on Swiss Info, obviously, but one of my favorite hobbies whenever I fly above the Alps in the good old days when we used to fly, was because of having grown up skiing mostly in Switzerland as a kid, growing up in Germany. My hobby has always been trying to figure out the names of the mountains from above, looking down from an airplane. And obviously that's a good analogy for the importance of taking a bird's eye view every so often, because when we do distance ourselves from our particular project and take a broader view, sometimes we're surprised how we can innovate and how we can see things that are patterns or distinctive features that keep reappearing and that may give us a hint into where we need to pay attention in the future. So I think one way that I like to approach these kinds of scenarios is really by asking questions that, first of all, sound very trivial, but actually aren't. And so this would be the first question, is sort of, what is the goal of all of this, right? So why are we developing drugs and vaccines? And obviously, everybody's so great to get a handle on this pandemic and to understand how we can bring our economy back and whatnot. But this is more a question of cui bono, right? What is the goal for the patient? What are we trying to do? Are we trying to alleviate symptoms? Are we trying to prevent transmission? Are we trying to prevent outbreaks? I'd just like to remind everybody to think about that question with each of their project. What is the ultimate goal for the end user? I'm not talking about now the broader scale, but which is your auditorium? Who is your end user? Who are your stakeholders? And what is the ultimate ideal outcome if everything works out the way you want to? And the answer to this question may be very different from person to person. Seeing there's okay, that's the best way to change slides here. So one way to take that bird's eye view is a technology I've learned in the past when I was working at Stanford. Many of you know that this was the first place where a D-School was created and co-funded at the time also by Hasso Plattner, who co-founded design thinking, the first two design thinking schools at Stanford and in Potsdam near Berlin at the Hasso Plattner Institute. Design thinking is one of the first kind of very formalized methodologies for innovation. And it basically leans on the thought patterns that you can see among product designers, really, and that's where this was implemented first. In thinking about breakthrough approaches to designing a new product or a new concept, whichever that may be. This is a picture of a project that we did in collaboration, so the Vienna Vaccine Safety Initiative did in collaboration with the University of Potsdam at that time, the school there, looking at specifically questions of disease prevention, infectious disease prevention. The teams are radically interdisciplinary, usually not medicine, but other disciplines, forced to find a common language, forced to disrupt. And we had a question about sort of, how might we enable, this is always the design challenge question that is defined ahead of time with the mentoring team and the faculty and the teams themselves. How might we enable parents and children to prevent infectious diseases was the design challenge question here. So we did not ask about vaccines per se. We really asked about any approach. And now COVID, many years later, this was in 2011 when we started our first D-School project, this has become blatantly obvious, right? So whether we talk about PPP, whether we talk about isolation measures, disinfectants for hands and surfaces, whether we talk about shortages of viral transport media, diagnostic capabilities, logistics of where people are tested, all the way to the way we organize healthcare around acute respiratory infections is not trivial at all, and in many ways insufficient to allow us to do the best possible with the tools that we have available currently. So basically, the idea is to really keep a circular motion. I'm not sure if you can see my pointer here. To basically go through a project where you, in a way that you keep going back to the last or second last stage and revisit that at the point when you already think you have ideated a new idea. You still go back to the very beginning of the observation, the point of view, and the ideation. But ultimately, you prototype relatively quickly and you test relatively quickly in order to really put the end user at the center of the innovation process. And we just heard the word implementation in the introduction. So that is then already foreseen when you start your observation. So that is probably one of the most striking components here, in addition to really focusing on the ultimate end user. And the way that is done is by the creation of a persona, like a typical user of your whatever product or concept or ultimate development that you're expecting, and to do very early research, can be qualitative or quantitative, of these persona types to understand which main insights you have to be aware of at the very early conceptual stage of a product or concept or model or whatever you're developing. Yeah. This is now the example of the vaccine or the disease prevention concept. We've done a number of design thinking projects throughout the years, but this is a very good example. You can get very early insights depending on how you pick your persona and what they're telling you at that point. There are other design thinking projects that may be dealing with airport security, for example, and that team went to Israel as one of the countries with the strictest airport security to understand where are the delays, what's happening, and to think outside the box. We've published some of that process, this particular project, and what we learned, and just giving you a few take-home messages. Of course, communication matters. This is not just for vaccines, but also for drugs. As you all know, especially the basic scientists among you, some of the more innovative products aren't entirely clear yet whether they will go the route of prevention versus treatment. There are antivirals that may have very good early use in post-exposure prophylaxis, for example. They are monoclonals, which in the US are handled like an antiviral by the FDA and by the EMA, more like a biological, but they're somewhere in that gray zone in between. We've seen this with influenza, and we'll see this again with COVID. We have vaccines that have a very different approach from each other. And the confusion is blatantly obvious. It's been blatantly obvious for a long time, but with COVID, I think it's been just adding to the overall confusion. Everybody's tossing around some words and pick them up on Twitter or somewhere, and it's really difficult to sort through this for the end user, right? The second thing is that timing matters. This is an example from the design thinking project where we found some interesting insight. If you think about vaccine prevention, and if you think about the classical way pediatricians get to know their teams or get to know their parents and families, let's say it's this young persona couple that we were talking about that has their first child. The first child may be the one where the decisions around vaccination are cast. But somehow, the first encounter with a brand new pediatrician usually happens in the most stressful moment possible, i.e. when nobody gets any sleep. Those of you who are parents will know what I'm talking about. Everybody has Olympic rings around their eyes and is sleep deprived and stressed out, and everybody in the family is weighing in on the topic during the dinner conversation. And now they have to take a decision on the first vaccination. This is the worst possible time point to sort through this. Plus, there are a lot of other things that have to be done during these early pediatric visits. The house has to be made ready. Everything has to be sorted out. So why not talk about this at a time when the parents are getting ready for the child during pregnancy? Why are we communicating this at this time point, which definitely doesn't work? So this is an example for very out-of-the-box design thinking innovation. A similar example that you're all familiar with in the respiratory virus field is the current neuraminidase inhibitors for influenza are given at a time and at a location where patients are often outside the window of time where the drug is most effective. Right? If we talk about academic tertiary care hospitals, until you're desperate enough to end up there, you're most likely outside that window. So these are examples of how do we have to figure out workflows and healthcare delivery around these issues. The timing matters also in terms of, sort of the dialogue around a decision that a patient has to make. This is not just when you do informed consent procedures and need to give enough time, which is important when you get an audit to show that you did. But also really in this long-term thinking when you partner with patients in routine care is to help them through these debates in a structured way. And this was like the idea of the baby mother pass add-on that would allow young parents to capture some of the rumors they're hearing about vaccinations in order to discuss them with their physician at the next visit, for example. All of this dovetails nicely with some work that we did sort of around the question of the importance of the patient voice in vaccination, vaccine safety. Are we actually listening? If we're not listening, why not? Are there time constraints? So if I develop a drug or a vaccine, how will that be communicated ultimately when it's available, when the package insert is ready, when everything is there? We've seen this with COVID vaccines. That's not trivial. Nobody quite understands why they're being offered one vaccine as opposed to the other and who took which decision. And patients have questions. Are we leveraging telehealth? Are we leveraging electronic means? Are we open to communication? Are we telling patients if they get a vaccine, for example, in case of an adverse event, as rare as it may be, what is your feedback loop? Who are you going to tell? And what is going to happen next? These are things that if they're unspoken, we also communicate something if we don't say something. That's a very important question. Again, important to anticipate during the phase of clinical research already. How does this fit in vaccination schedules? Is this too inconvenient to patients, and et cetera, et cetera. Next question is, of course, the question of signs and symptoms. So most vaccines and drugs, I would say, have in common that they're supposed to either mitigate or prevent signs and symptoms, right? Now, the question is, what is a sign and a symptom? And it's basically an abnormality, that's the official definition, that can indicate a potential medical condition, can indicate. All of us in the respiratory virus field know how difficult that is to differentiate different types of respiratory viral infections. Symptoms are considered subjective. This is now your patient and how they feel. For example, they may have fatigue or back pain, something that you don't even have on the radar as a clinician, but bothers them. Whereas the signs are the objective evidence of a disease. In other words, strictly spoken, the things that we would be picking up as a clinician. who assesses that sign or symptom is a critical question in clinical research and beyond, right? If you talk to regulators, there are basically three types of symptom capturing mechanisms. One would be the clinician, the next would be the patient, which is up and coming, as you know. And the third is the observer reported, which is very important in pediatrics and also in geriatrics, when you have relatives or guardians or family members reporting on the patient's condition. This influences your clinical outcomes in your trial. Because if you're not clear on what you want to know and whose opinion you want to hear, then it may get very wishy-washy at the end. How and when do we document signs and symptoms is a critical question. How are these data standardized? Are we sure we're avoiding ambiguities, et cetera, et cetera? As you know, whatever is captured in routine care is not extremely thorough, right? So this is an example of a patient, well, we know that they have influenza, and this is some body parameter going up and down. In this case, it's a body temperature, right? How much does that tell us in clinical research? Is body temperature the right outcome measure, or do we have to look at something different? You all know the so-called Ordinal Scale that has been proposed for a lot of these large-scale research consortia on COVID that has massive problems if you try to compare different sites with each other. Obviously, in the beginning, the peak and the end of a wave or a season, the criteria for hospitalization, ICU admission, availability of beds, attending physicians' preferences, things that are circulating in the community as viruses or rumors or whatnot, all these things influence medical decision-making. So remember, if we're talking about patient-centered research, and I can't state that often enough, patient-centered research does not mean my patient outcome is what other people do with my patient. It may be utterly convenient because these data are already available, right? Where the patient is located, on oxygen, off oxygen, ICU, not ICU, dead or alive, in hospital, not in hospital. But is this really the thing that the patient wants to improve? What is their experience of illness? What is their experience of success of treatment or prevention? If a vaccine is supposed to mitigate disease severity, how does the patient know that this has been happening? That's kind of the preemptive problem, right? If I prevented something successfully, how do I show that I did? These are questions that are here relevant. We've worked with bioinformaticians in Berlin for a long time. This is a question of, when we capture a lot of data, you can do an unidimensional symptom analysis, and this was like the body temperature here. It's whether pharyngitis is present and in how many people, and you can do this. We call this Swiss Alps graph. No offense, it's like something that goes up and down in a zigzag. That can please your bioinformatician very little, because ultimately your patient is a lot more complex. Patient is really a multidimensional matrix of signs and symptoms that are present or absent at any given moment. I'm not talking about prediction. I'm talking about a precise observation of the status quo at the moment of a digital assessment or clinical assessment. This leads me to a very important other question of clinical research is, can we make a clinical diagnosis? So a lot of, as you know, flu trials have been done based on clinical diagnoses of influenza, which was leading us down a difficult path down the road. Because these diseases can be confused with each other. And of course, if you have a virus-specific antiviral, it won't help another virus. So if you're too foggy in nailing your diagnosis and including your patients, then you may dilute the potency of your drug sort of in the results, because you may be treating things that are not the disease that you're looking for. This sounds very easy, but it's not as easy as it sounds on first statement, because these symptoms are very similar. And if you go down the route of how do we think patients present, there are also, popular media that have done beautiful graphs and models of how COVID presents, and now everybody looks at this, and suddenly flu doesn't seem to be on the radar anymore. or adenovirus or metapneumovirus or RSV or whatever you want to talk about. Well, that's a problem because we're creating bias in our diagnostic algorithms in many situations that has to do with funding of diagnostic services and all kinds of other things. These diseases are rather, similar in their presentation. And there's also a lot of catch-22. So if you say the most Google coronavirus question is it worse than the flu that was in the early stages of the pandemic? What do we mean by worse? What is worse to me? What is worse to my patient? What is worse to my hospital, to my clinical trial? These are very different answers potentially. The CDC has created that self-checker early on that created a lot of bias because people thought, okay, everything that's in here, and it was modified a couple of times, is related to COVID and nothing else. Whereas the things that are not in the self-checker have nothing to do with COVID. Of course, we as experts know that that's not true. Pretty much every symptom that was in the self-checker could just as well be another respiratory virus or ILI-inducing virus, to say the least. If we look at a certain stage, we had these symptoms and everybody was talking about new loss of taste or smell. And this was now the golden goose that we thought we had found that this would distinguish, COVID from influenza and other respiratory viruses. That, of course, it does not hold true if we start looking into it. I don't want to go into too much detail, but, the usage of Google Trends was used as a proof that people were starting to Google loss of smell during a COVID outbreak, but obviously this is a complete observer bias and a circular conclusion because if the CDC puts out that information, then people will start Googling it, not the other way around. So that was a bit problematic. And is loss of smell or sense of smell a new symptom? If you ask ENT doctors who've been dealing with this for decades and who also have the right testing methodologies to really distinguish and precisely nail down a diagnosis of a loss of sense of smell or taste, then you find a huge differential diagnosis of other conditions that can create this. First and foremost, common cold and ILI, flu and other things, but there are also a number of non-respiratory or non-infectious disease causes of this. So that's a problem. And if you go down sort of the decision tree of a medical history, you will have. This is from pre-COVID times. This is ENT literature telling you what to do with a patient who has a suspected loss of taste and smell. And you can see that this is much more complicated than a very simple medical history and that somebody checking a box somewhere, right? You have to really think about also serious causes of that sign and symptom. And I don't want to go into too much detail, but there's a lot of ENT articles, strangely enough, from Germany from decades ago, looking at influenza and anosmia. That's not a new observation. And olfactory disorders following upper respiratory tract infections in general. And obviously, patients with post-viral olfactory dysfunction have been described, including with pre-COVID human coronaviruses. And there's long-term serious olfactory loss in colds and flus. And in fact, if you vaccinate against flu, you can, have basically a decreased rate of issues with olfactory dysfunction in the following flu season. Well, that's kind of interesting. So that leads us to the next question. Can we distinguish the viruses sufficiently well, not just in the clinical research, but if we think about clinical trial design or vaccine design, looking at vaccination failure, what is the capacity after we have marketed a product, right? So if we nail the diagnosis really well, because we put a lot of money into it during the clinical trial, but then in actual real life, this kind of diagnostic capability, for example, is not available to the clinician who will now have to decide whether they use the drug or not, we will be facing a problem. And we did a lot of research on my team about can we distinguish respiratory viral infections based on clinical features. We've done a very, very detailed literature review looking at the major respiratory viruses And we've done detailed analyses on some of the common prejudices, I would say, in pediatrics, that fever and influenza are somewhat linked. You see that that doesn't hold very easily true, it's too fast, or fever and RSV would be less likely. That's also not entirely true. Another example is the rumor of wheezing being more indicative of RSV rather than flu, sort of, but not that straightforward. You know, if you look at these forest plots, they're all over the place and pretty much around the marginal point of no relevance. And the confidence intervals are huge, partly because of the study design problems. So what we did at the time in Berlin is we had a very large cohort of 6,000 children where we installed a quality improvement program with the Robert Koch Institute. And we use digital apps to do standardized disease severity assessments at the point of care that were very comprehensive. We started with 1,500 data points per patient observation, and this cohort was started in 2009-10. Then standardized risk factor assessments and using mobile applications at the bedside. Robert Koch Institute was looking at the major respiratory viruses in individual PCRs, where the primers were updated based on what's circulating in the population in Germany, which is very different from available diagnostic tests, commercially available diagnostic tests. And then we used culture-based microbiological analysis and MALDI-TOF. This is a very busy slide, and I just want you to look at the size of the confidence intervals here mostly. What you see is that, when we did the literature review, looking at tens of thousands of patients, because of the heterogeneity in how the literature was generated, the confidence intervals are very big, and we didn't find as many associations. So these are symptoms, and these are the major respiratory viruses, right? So large confidence intervals and fewer associations were discovered. When we looked at our cohort, where at the point of care, we captured every single data point and ascertained it, whether it's present or not, regardless of what is in a routine medical record. So now you suddenly have to look at your patient, which is always a good idea, in my opinion. Now you suddenly have much more narrow confidence intervals, even though the total number of patients is a lot lower. And you're going to be detecting things and identifying new signals if you do this well. And this is sort of a matrix view of the situation. So where we have a number of clinical signs and symptoms, you don't need to read them all. And then we have these different viruses, and we always compare here the literature in the left column and the cohort on the right, and the literature cohort, and the first one is flu, next is RSV, and so forth. In green, in dark green, we marked those combinations of literature and cohort that had odds ratios pointing in the same direction and being both significant. Okay, so here the literature and the cohort were in agreement, and there were quite some examples. For example, here with cough and flu. But, disappointingly for us clinicians, that is just as true for cough and RSV and cough and human metapneumovirus. So just because the patient has a cough doesn't mean you know what they have. And if they have a cough, it doesn't rule out a seasonal coronavirus, which we looked at, or Boca virus or something else. It can happen. It just isn't that significantly correlated, right? Orange is a negative correlation. So we found that only in a significant way, and only in myalgia and human rhinoviruses for some reason. This is also something that we as clinicians don't think. That's what a bioinformatician does, but we don't really think like that, right? We don't say this patient does not have myalgia, ergo it's more likely that they have virus X. But that's something that we can do with AI and more complex analyses, which is what we're working on right now. So this is interesting to see, but the conclusion here is you can't rule in or out just based on the symptoms. You need diagnostic testing, and we're currently doing decision tree analyses and very complex AI analyses to see how close we could get with a perfect decision tree based on symptoms alone versus a simple antigen, a clear waved antigen test versus PCR. And you will see that diagnostic testing is always superior to whatever the smartest AI-supported clinician could come up with. And so the question now for the patient is, what if I do get sick? And here, by the way, that I didn't want to forget to mention is that we did see For coronaviruses, we did see some association with conjunctivitis, which I thought was quite interesting because that has been picked up in COVID as well. So this kind of potential capillary syndrome, and I know that there are people looking into this, and this research program might be of interest. What if I do get sick now? The patient gets sick, what's next? And here's important to also think about the bigger picture of antibiotic indications and resistance, and soon also antiviral indications and resistance. There have been shocking studies showing that, people don't really still know what's happening when they don't get an antibiotic. I'm preaching to the choir here, I'm sure. And then the questions like how patients' experiences affect healthcare seeking behavior. These are all pre-COVID questions, right? And you can see that our perceptions as patients of what we think our disease may entail, the things that we find out via Dr. Google or asking our neighbor or friend or family member influence our healthcare seeking behavior. And this creates bias in your clinical trial. Be aware of that. You're not getting a random sample from the population. You maybe get the most nervous people or the most easygoing people, or depending on your trial design. So these are very important questions, and you can see how people differ in their attitudes toward getting medicine, taking no measures at all, depending on the duration of illness, etc. So even throughout the illness, these things change. This is of critical importance for those of you who work on antivirals, because there may be very similar trends in antivirals, like how willing are people to start a new drug depending on how long their disease has been going on, etc. So the next point is which symptoms constitute severe illness to the doctor versus the patient. These are different things, right? So what does the doctor think is a severe illness? What does the patient think? We started in our cohort with very specific assessment and these apps that I mentioned before, where we really did very, very deep literature analysis, again, looking at all symptoms that were described in the literature in clinical trials, non-randomized and randomized observational studies, epidemiological studies, symptoms in relation to respiratory viruses, to try and see which ones are considered severe and to develop a severity score that will help us nail down the clinical picture at any age group. So we started in children, extended it to adults, in any age group at the point of care as they are presenting to us, regardless of which respiratory virus they're affected by. And you can look this up in the publication that I mentioned. This is the idea of the app, which will now be used also in a European project in a consortium that we're going to be leading beginning May 1st, which is literally this weekend. And where we're also collaborating with the Finnish Public Health Institute on some of their COVID logistics and surveillance, using severity scoring at the point of care as an additional tool for multidimensional mapping. We've been doing the proof of concept in the United States and very low-key community clinics. We set up the first ever project where we do real-time mapping of influenza incidence and severity, influenza, RSV, and now also COVID. And you can find some of the early papers here. Remember that, and that was very interesting for us to see about the score. When we looked in the initial cohort, a lot of people got antibiotics, way too many. Looking at the up to 1,600 data points per patient observation that we have been gathering, we asked the bioinformatician to tell us which data point is most likely correlated with antibiotic treatment decision. And it turned out to be our severity score, which is interesting because the clinicians were blinded to the score. They had no idea what the score was about. That was done by an independent quality improvement team. So that's quite interesting to have something that relates to the physician level of anxiety. Whether giving an antibiotic is the right decision or not is a whole other question. So we developed sort of SOPs that would help to minimize and cut the use of inappropriate antibiotics in half, and also cut costs in half by doing things like severity scoring, review of the vaccination record, and targeted diagnostics. Remember, when you develop a drug, that the duration of illness is not the only important parameter, but the overall severity. And that's been something we've a hard lesson to learn from Tamiflu, where the focus was lots on how much do we shorten the course of illness. Remember that we may also have the same duration of illness, but the area under the curve may be a lot less, which may make a big difference to our patient if they're able to function or take care of themselves as opposed to, needing other people, etc. And in this respect, when we were collaborating with regulators and starting to work on a patient-reported outcome of our severity score as a patient-reported version, we thought, okay, great. Next thing we're going to do is we look at the literature and we see what matters to patients, not just doctors. Well, to our great surprise, there was no information in the literature on that. So we decided to do this research ourselves. And this is the Symptom Survey, which we've developed in collaboration with patient organizations to really learn what matters to the patient in COVID, flu, and related illness. And what do they want to see treated by an antibiotic or antiviral? They usually don't distinguish, but we know what, let's say, antimicrobial, if we talk about respiratory infections in general, and which signs and symptoms do they want to see prevented through vaccination? Is that the same? Are they different, etc.? So we have started this project currently mostly in Europe and the United States. This will be expanding more internationally. And, this is an example, if you'd say, so how important is the symptom to you and how you feel and function? That is what interests us, both for an adult talking about themselves, talking about their child, talking about their elderly family member. These decisions may again differ depending on who you're talking about. This is a poster that we presented last year during an ICF antiviral group meeting. And, I just wanted to emphasize the importance of patient-focused outcomes here. Don't forget to ask your patient how they're doing, as trivial as it sounds, because these are your end users. And if they're not helped, then they will not accept whatever we come up with. And this is very important. So then the next point is data standardization. We've worked with CDISC very intensely in the FDA on those kinds of questions. Don't forget to remember that data formats and data standards are of critical importance in order to pool large amounts of data in clinical research, to share it, not just within, the Swiss system, so to say, but also with international world of science out there. And again, we all just learned that this is not just a respiratory virus. Do patients know that? Are we biased? What about nausea and diarrhea? What about heart attacks, damaged kidneys? Those of us who have been very deeply involved in influenza clinical care know that all too well, that some of the cases we see in the ICU may be presenting primarily with cardiac issues, rhabdomyolysis, and some other non-respiratory symptoms. Not everybody ends up in ARDS, sometimes in combination, but not always. So does that mean we have to communicate this differently? Does that mean we have to prevent different things through vaccination? Does that mean we have to change our CRFs in clinical research? I leave these answers up to you, but it's just food for thought, looking from the outside in. And again, this is just an invitation to please collaborate. We'll be happy to facilitate any of the work that you do. Also opening the line of communication, and that's why I put my email contact here and the Twitter also of our organization. Think about the patient first, and then you will succeed. That's, I think, my most important take-home message here. Thank you very much.